Hi, I'm Jessica Houtz. I'm an editor with Cell Reports, and I'm speaking with Itamar Harel, who's a professor of genetics at the Hebrew University of Jerusalem. Thank you very much for joining me.
Thank you.
So you had, started your talk sort of talking about the challenges with aging research and that there's sort of this rate-limiting aspect of how long it takes to study these model systems. And so you spearheaded an initiative to sort of develop the killifish as a strong, tractable genetic model for studying aging. So could you talk a little bit about why you did this?
Yeah, so I started my PhD with genetics in mice, developmental processes like heart development and facial development. And at some point, it seemed to me that during development, fixing something, it's almost too little too late, because once a mutation happens, it's going to mess up things quite badly. But with aging, it's a process which is really long and has inputs from the environment and from genetics and from a lot of aspects, and there's a lot of opportunities to manipulate it. So that's why it was really fascinating to me. And the killifish was because I really wanted something that I can manipulate in the lab. With mice, usually you outsource to make lines, but with fish, you can just, you put them together. They have a beautiful dance, actually, and you can collect the eggs and inject CRISPR, and you get a new line. So this is what I started doing when I joined Anne Brunet's lab, and I really wanted to transform them into a genetic model. Otherwise, I would have done something else, and it worked. So I think since then, starting my own lab, this is our focus, trying to really tease apart the genetic processes which can either slow down or accelerate aging.
Yeah, so these fish have quite an accelerated aging process that is condensed into this short time frame.
Right.
And that kind of enabled you to take a look at a particular question about, this correlation that you see with the age of maturity onset with the lifespan of these animals. So can you talk a little bit about what has come out of that, what you have noticed with those fish?
So I see it not as accelerated aging, but more of a compressed life history.
Okay.
Because everything happens proportionally, like the onset of puberty and adulthood and eventually aging happens in a very similar way to us, just everything is compressed proportionally. So it's accelerated aging usually has association with a pathology, like telomerase disease or others. So they age in their own pace, just like Drosophila ages really fast, and mice, and then us, and then the Greenland shark, which does it in 400 years.
Yes.
So we focus on really try to identify how we can scale this process, very similar to what was done across evolution. So we try to manipulate the onset of puberty and delay it or accelerate it, and try to see what happens if we really can do the same thing for the entire life history. And this is what we did. We delayed puberty and accelerated by manipulating a specific gene and two isoforms of that gene. And we were able to not only accelerate puberty, but also increase growth and also increase age-associated diseases like melanoma. So all together, if you track all of this together, it's really compressing even further something which is on the extreme side. So overall, currently, killifish are the fastest or the most compressed life history across all vertebrates. So we were able to do it a bit further.
That's, very fascinating.
Yeah.
Can you talk a little bit about the sort of balance of resources that you discussed are sort of at war there with the reproductive capacity versus somatic maintenance sort of argument that's going on?
Yeah. So this is another work that we published last year, and something really cool that, it's a fun fact, that Korean eunuchs that were, I think, about the 1800s lived 15 to 20 years longer than expected.
Oh, wow.
Yes. So it shows you that this link between reproduction and aging is not only like something theoretical, but it's also something related to us. I'm not willing to get castrated, so the idea is really try to identify how reproduction controls our body overall. And there's this notion that the immortal germline, which almost passes from one generation to another, might have evolved ways to control the carrier, which is us. So we tried to really investigate this resources. We really tuned down the amount of resources that the body can fuel to the germline. So all these fish were sterile, but they had a reduced amount of germline development, all the way to actually losing the germline altogether. And we could see that if you do that, the resources didn't seem to have any effect, because if you remove the amount of resources invested, nothing happens. Only when you remove the germline altogether. And there you get all these cool benefits. The females get a superpower. They become resistant to DNA damage, radiation, and their tail grows much faster following this stress. And the males live 50% longer. So now we are developing this mouse model based on this, trying to really identify what are the mechanisms which might be conserved across vertebrate species.
What do you think is underlying this sex difference in the responses you see, this super healing in females, but you see this longevity effect and metabolically programming in males?
So I don't know why every intervention we've done so far was sexually dimorphic. And I think some key aspects are linked to evolution, because if you perturb something related to reproduction, then another pathway which is linked to this is sex hormones. And sex hormones like testosterone and estrogen are tightly linked with this entire process. So if you disrupt them, you will get a sexually dimorphic phenotype. So this is one thing. Another thing that some of our recent work showed, that if you fine-tune metabolism, you also get lifespan extension, which was mostly, in our case, in males, because we really perturb a specific enzyme that controls synthesis of ATP. But another thing, I think it was mentioned throughout the conference also, is that some aspects of metabolism, even though it actually gives rise to longer life in males and females, it's through different responses to fasting. So if an animal, an organism, even us, fast, so females and males will turn up or turn down different pathways, even to reach a similar consequence. So that's why we think that the effect could be also sexually dimorphic. But I think most of the things we really don't know yet.
Well, it'll be fascinating to see how that evolves over time.
Thanks. Yeah.
Well, thank you very much. I appreciate the conversation.
Thank you. It was fun.
All right.